Good morning, good afternoon, good evening. We're broadcasting live this afternoon from Dunedin, New Zealand. I'm Phil Batterham. I'm the Secretary of the International Genetics Federation and hosting the seminar today. I'm in Melbourne, Australia, and I live on the lands of the Wurundjeri people, who are our local First Nations people, and I'd like to acknowledge them, their leaders, past, present and emerging, and would urge you to consider the lands upon which you live and the First Nations people there. Before we begin, just a couple of announcements. You can see a slide about our next Asia Pacific Genetics Seminar in November, featuring John Bowman, who'll speak about the origin and evolution of land plants. And I also want to mention the upcoming 23rd International Congress of Genetics and Genomics, which will happen in Melbourne next year. This Congress happens once every 5 years and first started in 1899. It'll be an extraordinary event. So I draw it to your attention. And now I'd like to hand you over to Stephen Leslie, who's going to introduce today's speaker.
Thank you, Phil. It is with great pleasure that I introduce Professor Lisa Matisoo-Smith, Professor of Biological Anthropology in the Department of Anatomy at the University of Otago. Lisa is a bioanthropologist who uses contemporary and historic materials to understand the prehistory of the peoples of the Pacific, a field to which she has made major contributions. Her work focuses on Pacific peoples themselves and their commensal plants and animals. She utilises both ancient and modern DNA methods to answer a range of anthropological questions regarding population histories, dispersals, and interactions. She is also interested in how human history and population origins may be related to some of the health issues facing Pacific populations today. Importantly, she is committed to working in close collaboration with Pacific communities. She has received many accolades for her work, including being elected a Fellow of the Royal Academy of New Zealand in 2013 and winning the Academy's Mason Jury Medal in 2018, awarded annually to New Zealand's preeminent social scientist. In this case, for her groundbreaking work that has, through strong relationships with New Zealand's Indigenous people, reshaped our understanding of the last great human migration into the Pacific. She's also a Fellow of the Society of Antiquities from London, and was the principal investigator for the Pacific Islands with National Geographic's Genographic Project. Today, I'm delighted she will share some of her work with us in her talk entitled The Last Great Human Migration, Recent Advances in Understanding Human Settlement of the Pacific. Thank you very much, Lisa.
Kia ora, thank you. And let me just get my screen sharing here. Oh. Assuming that's all looking okay. Okay, ngā mihi nui kia koutou. Ko Surmunang Magi, te maunga o Okutupuna, ke te repo o Vasilina, Estonia te wai o Okutupuna. Ko Andres Matisoo tōku mātua no Estonia ia. Ko Julia Moores tōku whaia no America ia. Ko Brent Smith Takatane no Tamaki Makaurau ia. Ko Lisa Matisoo-Smith tōku ingoa I whanau mai ahau ki te motu Oahu, Hawaii. Engari I tipu ahau ki te marumaru o Maunga Fuji, Hapane. E noho ana ahau I Aotearoa mō ngā tau torutakau mā rima. No reira, te moana nui a kiwa ke roto tōku ngākau me tōku wairoa. No reira, tēnā koutou, tēnā tātou katoa. So I have just given my pepeha, my background, my description of who I am in Te Reo Māori. And what I basically said is, I think what a lot of people wonder about when I start talking. So I am, my family, my father is originally from Estonia, and that is kind of where I consider my real homeland. My mother is American, but I was born in Hawaii. I grew up in Japan, and I have lived in New Zealand for 35 years. So the Pacific Ocean from Hawaii, Japan, also California, and now Aotearoa, New Zealand, is very much in my heart. As a biological anthropologist, it's also in my bones, I can explain, and the isotopes in my bones and my teeth. But it's in, it is what drives me. So giving this talk today to this group of people also links the Pacific Ocean with Australasia and with Japan. So I spent 10 years living in Yokohama, well, Yokosuka in Yokohama, Japan, from 8 to 18. So yeah, hajime mashite, dozo yoroshiku. So I'm going to kind of take you on a rapid 60,000 years of human history, but and describe some of our own research of my group here at the University of Otago, and colleagues that I have around the Motu, around New Zealand and around the world, but also talk about some of the more recent research that's being contributed by labs around the world, and is helping us understand the settlement of the Pacific and raising some new questions for us. Now, let me just see if I can move that so that, there we go. Maybe that'll work better. Okay, so just to kind of get people into the time frame, and I should have checked, there we go, that we're talking about, and place us all geographically. When we talk about the settlement of the Pacific, it's a fascinating, the Pacific is really quite central in that we have some of the very earliest out of Africa migrations, and the last kind of major migration in terms of the settlement of the Polynesian Triangle. So I'm going to be focusing primarily on that later settlement, but I will talk a little bit about the history of the early settlement of the region, because that is also quite important. But this is kind of the Pacific as most people who are familiar with the Pacific kind of think of it in these definitions that were initially proposed by D'Urville in 1832, one of the European explorers. And that kind of breaks the Pacific up into three broad regions: Polynesia, Melanesia, and Micronesia. And of course, these are very historic and somewhat problematic, somewhat racist in some contexts, definitions that early Europeans made, recognizing quite a lot of linguistic and cultural similarity within Polynesia, basically translated as many islands. The region of Melanesia, or the dark islands, describing the darker skin of the inhabitants, from New Guinea through the Solomon Islands, New Caledonia, Vanuatu, Fiji. Fiji sits kind of in between Polynesia and Melanesia. And then the islands of Micronesia, the little islands that are north of the defined region of Melanesia. So we tend not to use these terms when we're talking about the settlement history, but they certainly still reside in people's general references and knowledge and understanding of the region of the Pacific. But Polynesia and Micronesia, we still use from biological terms. Again, they are not as precise as many people would like to think. But yeah, we tend to think of the Pacific in different terms, which I'll present now and that change through time. So when we're talking about the initial settlement of the Pacific, what the landscape would have looked like when the first people arrived, of course, sea levels are much lower than they are now. And we have New Guinea and Australia, Tasmania joined together in the greater landmass referred to as Sapu. and what is now island Southeast Asia. Much of island Southeast Asia was joined to the Asian landmass in the continent that we describe as Sunda. But Sunda and Sahul were always separated by the deep water channels in between the area that we refer to as Wallacea biogeographically. And so we see, of course, the classic biological differences between the Asian mainland and Sahul, the Australian flora and fauna. But so people could have walked basically from Africa through to Sunda, but in order to get to Sahul, they required some form of watercraft. And even at the periods of lowest sea level, this would have required crossing multiple gaps of a minimum of 70 kilometers. So, you know, early kind of ideas of how people got to Sahul sometimes suggested what we might consider ridiculous things like the pregnant woman hanging onto a log, being washed out to sea and landing on the shores of Australia. And that was the source of the Australian population. We now know that that is not the case, and that there is evidence for significant, probably multiple colonization events of small hunter-gatherer populations arriving on that continent, probably or potentially on what is now New Guinea and also on the Australian mainland. Archaeologically, we have evidence of this. We've known for a number of years, based on early mitochondrial data, but also on linguistic data and archaeological data, that the settlement of Sahul was very ancient. But research in the last 10 years or so has really been pushing back the date of initial human arrival in Australia. And Clarkson et al. Suggest from the site of Majid Bebe that the earliest dates are 60, 65, and possibly even earlier, 1,000 years ago. We also have significant evidence in the New Guinea highlands of occupation by 50,000 years ago, and by 40,000 years ago, archaeological sites are dotted around the continent of Sahul, indicating that people spread very, very rapidly, probably following river coasts and riverine systems. And we do see some genetic variation, which indicates that there was genetic isolation, probably related to distance and the highlands of New Guinea. So we have New Guinea-specific lineages, mitochondrial lineages, for example, and Australian-specific. But I'm really going to be focusing today on the settlement of what we call remote Oceania. So during the Holocene, so around 11,500 years ago, sea levels rose to what is close to the sea levels that we see today. And so New Guinea became separated from Australia, as did Tasmania from the mainland. But note that the islands there in the Bismarck Archipelago were never attached to the mainland of Sahul. And we have occupation of those islands in the Bismarck Archipelago by 40,000 years ago, and settlement of the Solomon Islands by 30,000 years ago. So this area that has this deep history, this deep ancestry, really extends out through the Solomon Islands proper. So we see significant genetic variation, we see significant linguistic variation, deep history, and people living out here at the time that really Homo sapiens are moving into and competing with Neanderthal in Europe. But people don't move beyond that line drawn there, B, into what we call remote Oceania until much more recently. And this represents what we like to refer to as the last major human migration, the last migration out to lands that had not been settled. So we'll be talking really quite a lot about Near Oceania, remote Oceania, very deep time and history and variation in Near Oceania, and much more recent in remote Oceania. Now, we think that this line that we designate between Near and remote Oceania relates to kind of voyaging skill, experience, technology. When you're sailing within Near Oceania, you're always sailing within sight of land. So if you're sailing from one island in the Bismarck Archipelago to the next, with the exception of Manus, which was settled about 20,000 years ago, you can always see the next island before you lose sight of the island that you left. So psychologically, you're going from point A to point B, a known point. When you go beyond the Solomon Islands proper, these are, throughout this, these are high islands. When you move out into remote Oceania, you're talking about much greater distances and a large number of low islands. So people, in order to reach these islands, have to sail out of sight of land for significant periods of time. And so we think it was both kind of a psychological barrier and also related to kind of improved voyaging technology, so use of boats and sails. And, this area of Near Oceania and island Southeast Asia has been described as a possible sailing nursery where people were learning these skills. At about 3,500 years ago, they had the skills and the technology available to sail beyond that kind of psychological barrier. So the settlement of remote Oceania, the first people to arrive in remote Oceania, are associated with a Neolithic expansion that probably began on the Asian mainland with the development of rice agriculture, population growth and everything that goes with that, driving an expansion of people out of Taiwan who speak languages that belong to the Austronesian language family. They moved through island Southeast Asia, through what is now the Philippines and into Indonesia, and then down into Near Oceania, where they interacted with the people who had been living there for tens of thousands of years. And then they moved from there out into remote Oceania, reaching the islands of Vanuatu, New Caledonia, Fiji, and as far as Samoa and Tonga. So you can see the distribution of the Austronesian languages. We see the most linguistic variation in Austronesian languages in Taiwan. So Taiwan is identified kind of phylogenetically as the homeland of the Austronesian languages. And then you can see the breakdown of kind of cladistic breakdown of the languages, the Austronesian languages, into the Central Eastern Malayo Polynesian languages, which are spoken through island Southeast Asia and into Madagascar. And then the yellow, which we're really going to be focusing on for the rest of this talk, the oceanic subgroup of Austronesian languages, which are spoken by the people, most of the people of remote Oceania, and some of the people of Near Oceania. And there are a few kind of unusual ones that we might be able to talk about later. So that's the linguistic evidence. We also see this expansion, this Neolithic expansion. These are farming people with domesticated plants and animals, and we can identify them by an archaeological signature as well. And it is what this archaeological signature, archaeological culture, we refer to as the Lapita cultural complex. And so we see the appearance around 3,350 years ago in the Bismarck Archipelago of a number of new features that are very clear archaeologically. We see the introduction of a number of plant and animal species. These people used what we call transported landscapes. They carried their domesticated plants and animals with them. But we also see a number of artifacts and settlement patterns and so forth that go with this Lapita culture, and we can track this through the Pacific. Most notable and obvious, again, being a Neolithic or domesticated people with domesticated animals and plants, and pottery. That's the other kind of classic archaeological signature of a Neolithic culture. And so we see pottery appearing in the archaeological record for the first time in this region associated with Lapita. And it is this particular shape and forms of the pots, but also a very identifiable way of design and decorating those pots that we call dentate stamped. So it's like taking a little comb and impressing into the pottery some very specific but very intricate design patterns. So these decorated Lapita pots, we think are primarily ceremonial. They were also using undecorated and plain pots as well. So you always have to remember, but there are a full range of artifacts that are associated, shell artifacts, bone artifacts, and as I say, the domesticated plants and animals that people carried with them. So that's how we identify them archaeologically. Well, again, reminding people that language, biology, and culture do not always go hand in hand, as I am sitting here speaking English with an American accent in New Zealand. So we always have to think about that. So this shows the basic timing and extent of the Lapita expansion. You can see it was remarkably rapid archaeologically. Within a few hundred years, people had moved from the Bismarck Archipelago all the way to the edge of the Polynesian Triangle, reaching the islands of Samoa and Tonga, about 2,900 years before present. So this is the archaeological use of BP, not base pairs. It's years before present or years ago. So first 3,350 BP years ago in the Bismarck Archipelago, and rapidly expanding to the edge of the Polynesian Triangle and Samoa and Tonga. Now, sailing out in this direction is quite a remarkable feat. Not only are they sailing for many, many days out of sight of land, but we've come to realize through voyaging simulations and sailing trips and various studies that there was a real strategy to the way that that Lapita peoples and their descendants voyaged and in a sense surveyed the Pacific in order to find these islands. Now, as you're moving into the Pacific, across the Pacific from west to east, you're sailing into the wind, and that is the more difficult direction to sail. And anybody who sails or dives knows going into the wind is hard. You do that first, you turn around and come home with the wind if you can, or with the currents if you're diving, because that's safe and you know where you're going. So we think that this is what indeed Pacific people were doing. They were engaging in a systematic exploration of the Pacific, go out for a couple of days in a particular direction. If you find land, you know where you're going, you turn around, you go home, and then you take your settlers with you. So you have a kind of difference between your explorers and navigators and your settlers. And that if you don't find land, the next time you go out, you go either a slightly different direction or slightly further, and say you don't find land, you can still turn around safely and get back home. You know how long it's going to take you to get back home, and you know where home is. And so Pacific peoples were using stars and other indicators, other natural indicators for that navigation. So this is a system of safe return voyaging. These are not people hoping to, pushing off the beach and hoping to find land and, dying at sea, which is often kind of the mental picture that Europeans had of how the Pacific was settled. So there's systematic and planned settlement, no or little loss of life, and difference between exploration and settlement. And we also have archaeological evidence of long-distance trade and exchange. And we see this in the Lapita culture, where obsidian or volcanic glass from near Oceania is being taken and traded all the way out to the edge of the Polynesian Triangle. And we see the continued use of these shared design motifs for quite some time, or for at least a few hundred years from initial settlement. So we also can see, genetic contributions to this general picture over the years have improved. This was an example from Jonathan Friedlander's work in the Pacific, looking at mitochondrial DNA variation. And we see the deep kind of ancient mitochondrial lineages, P, Q, and some of the M lineages that are found almost exclusively in near-oceanic populations. And then we see the mitochondrial B lineages that are East Asian derived lineages that appear to be associated with this Lapita expansion. And we see a mixture. If we go out into remote Oceania, of a mix of these near oceanic lineages, these dark lineages, and the B lineages that we think are carried by the Lapita people, this Austronesian expansion. And by the time you get out here to the edge of the Polynesian Triangle, there's very little, or so we believe, very little genetic variation, almost exclusively mitochondrial haplogroup B, and in particular, the B4A1A lineages, for anybody who's a mitochondrial DNA person. So the settlement of the rest of the Polynesian Triangle, say Lapita people got out here to Samoa and Tonga about 2,900 years ago, and the settlement of the rest of the Polynesian Triangle doesn't occur for nearly 2,000 years, 1,800 to 2,000 years after Lapita people arrive in Samoa and Tonga. And that's always been a question that's challenged archaeologists. Why did Lapita people stop sailing in West Polynesia? We certainly see that Lapita, there are numerous Lapita sites in Tonga and Samoa. There are a lot fewer. They're quite ephemeral. And so whether this was a population, kind of running out of steam, running out of people, it was very, very rapid. We don't really know. But what's interesting is when people begin moving back out into the rest of the Polynesian Triangle, they move into what we call Central East Polynesia, the Society Islands, the Cook Islands. So that's Tahiti, Rarotonga, the Tuamotus, and ultimately up to Hawaii, out to Rapa Nui or Easter Island, and finally down here to Aotearoa and out to Rekohu after that. And some debate as to that movement, but one of the interesting things is that people did not take pottery with them. Discard, get rid of whatever sorry about that. I thought I'd closed everything, so I wouldn't have that happen. So yeah, people did not have pottery. So why Lapita people lost their pots, why pottery disappeared, has always been a bit of a question for archaeologists and such. Now, just to give you an idea of the distances for people who are not familiar with the Pacific, and it just appears to be this kind of big blue space. Here is a map of French Polynesia overlain on the map of Europe. So the islands of the Marquesas up here in Scandinavia, the equivalent, the Austral Islands down here, and along the Mediterranean, and from the British Islands all the way across to nearly the Black Sea. That is the distance of French Polynesia. And if we can put that on the map, that is just this area here in the circle. So, these are significant distances that these Pacific people are traveling, both in the initial settlement, but also in the evidence that we have trade and exchange within the region over significant periods of time. So this required very impressive navigational skills and sailing technology. So until recently, archaeologically, linguistically, and genetically, we had this kind of picture, this story about the settlement of the Pacific, and it often focuses on the origins of the Polynesians. And so we always say Polynesians actually weren't Polynesian until they got to Polynesia. But it was this story that the Lapita people, who are a mix of Austronesian and Papuan peoples, so while they're in near Oceania, they're picking up mixing with the people who had been there for tens of thousands of years. And then those people settled, went beyond near Oceania, out into remote Oceania, all the way to West Polynesia. And then during those nearly 2,000 years, that's where Polynesian language, society, and culture developed. And then it's, for some reason, about 1200 or 1000 BP, a subset of these people went on to settle East Polynesia. And the argument always was that there's little genetic variation in Polynesia due to these bottleneck events. So that's the kind of standard model. When I started working in the Pacific, I was interested in trying to better understand the settlement of the Pacific, and in particular, the settlement of Polynesia. And could we use genetic methods to try to track the movement of people through the Polynesian Triangle and identify, for example, the origin of the Maori, the New Zealand Maori, and where did they come from? We knew linguistically and archaeologically that they're coming from Central East Polynesia and through oral traditions, but could we identify specific locations? Now, at the time in the 1980s, there was certainly a lot of mistrust of geneticists for a very good reason, given our history of sampling, seeing people as samples and taking those samples away and doing what we wanted with them. It was very clear to me that I was not going to be able to use human genetic data to answer the question. And I was very lucky to meet a colleague who introduced me to the fact that Maori, New Zealand Maori, and Pacific people more broadly introduced a number of animals and plants, say, to Aotearoa, one of those being Rattus excellens, the Pacific rat. And I realized that if we could figure out the origin of this rat species, that would give us an indication of the origin of the canoes that brought them, and therefore the origins of the people. So we could use the mitochondrial DNA variation. This was late 1980s, just after Alan Wilson and team had published their mitochondrial Eve data, that we could use this approach to study the commensal animals. And I decided to use the Pacific rat, one, because it was the one animal that got everywhere that Pacific colonists visited and landed. It's in the earliest archaeological sites, the remains, and it is a separate species to the rats that were introduced by Europeans, Rattus and Rattus norvegicus, so it does not interbreed. So the rats that we see on the islands today theoretically should be the descendants of the rats that were introduced by the first colonists who brought them with them in their colonizing canoes. So kia ora to Mary Roberts if she is watching, but this also resulted in my being introduced to a number of Maori colleagues who were kind enough to share with me their traditional knowledge and make me aware of the importance of the kiore, as it's known in Maori, as a cultural item. So in New Zealand, kiore are considered taonga or treasures. So a very different relationship with rats for Pacific peoples than Europeans, for example, have with rats and the association with disease and the plague and all of that kind of stuff. So as an example, here is a carving from the meeting house of the marae, at the University of Auckland, Tane Nui Arangi, of Ruanui, who's one of the navigators, the traditional navigators, ancestral navigators, with the kiore, with the rats traveling on his shoulder. So, and there's a lot of information about the importance of kiore in Pacific communities across the Pacific. And the fact that these rats were not stowaways, as is assumed, because that is the relationship with European rats, but these rats were brought intentionally as an important food item. And here's an example of the type of container that they might have been transported in, a little carved. The rats would be kept in here, kept away from the other food items that people are carrying with them, and they're keeping an eye on those all the time. Rats also, kiore, don't like wet conditions. They hate to have wet feet. So again, not typically going to be found in a canoe, climbing into a canoe. We also know that the rat is good food. I have indeed eaten kiore. It's quite tasty, tastes like chicken, but it was also an important food reserved for certain people, but it could also be a famine food. So depending on whether it was needed, but it was also important for gifting. And here's some kiore preserved in the fat in this gourd here, which is one of the things that Pacific people, Polynesians, and other Pacific people used instead of pottery in order to transport liquids and cook in and so forth. So doing this work, this commensal work, really brought me very early on to some very important lessons that I learned, and I've been been very lucky that happened early, and benefited my research from that point on. First of all, it was important that we consult, and for legal reasons. In New Zealand, we have the Treaty of Waitangi, and there was a submission to the treaty claims process for ownership over Indigenous flora and fauna, the Y262 treaty claim. So there were legal reasons, but also ethical reasons as to why we needed to consult with the Indigenous people where we were working. We set up consultative committees and trying to make sure that there was good consultation. This meant that we were discussing things like ownership of knowledge, who owns the DNA, for example, that we're generating, publication of data, interpretation, partnerships, and of course, relevance to Māori. So this was in the early and mid 1990s that we started having these conversations and building these relationships, which were absolutely essential for any work, but certainly work with Indigenous peoples and important flora and fauna. So this was the research that I did for my PhD. We showed that with the Kiore, looking at extant populations, mitochondrial DNA variation, that we could indeed use them as a proxy for tracking the movement and the origins of Polynesian populations. We then went on to look at a number of other commensal species, the 4 commensals that were carried by Pacific people, dogs, pigs, rats, and chickens, and then a number of plants as well. And with these other species, dogs and pigs and chickens, we relied exclusively on ancient DNA, because the dogs and the pigs and the chickens that have been introduced by Europeans and others are the same species, so they have interbred with those original populations. And what we see is that they're found, if at all, at very low frequency. The genetic markers of those initial introductions are not really seen widely in the populations of these animals today. But what we found was, surprisingly, it looked like there were at least 2 introductions of rats from island Southeast Asia into the Pacific. We saw multiple introductions, or we've seen multiple introductions of dogs. And strangely, dogs don't appear in the Lapita sites, so there's a bit of a debate about that, but they certainly don't appear through remote Oceania until about 1500 BP. So they don't look like they're being transported by Lapita people into remote Oceania. And we see evidence of 2 chicken introductions into the Pacific from island Southeast Asia. And one of these also appears to post-date 1500. So it looks like we're getting different populations from the same source region, but later in time. So this really led to the question, where are these second animal introductions coming from, and what's happening at 1500 BP? Something interesting is going on there. So this made us really kind of start to question this initial kind of the assumptions that we made about Lapita origins for East Polynesians, that there might be something else going on, and something going on that's not Lapita associated, and that's happening perhaps around 1500 years ago. And having conversations with other colleagues, archaeologists and geomorphologists, we realized in reconstructing many of these island environments at the time that people are settling, that there was a final settlement of sea levels. And this is, if we're looking at the Pacific, 3,000 years ago, these are the high islands of Micronesia that are exposed and where people can potentially live. But at about 2,000 years ago, the sea level drops at least a meter and a half or so, and exposes all of the atolls of Micronesia. And this basically presents kind of stepping stone islands of uninhabited islands that lead directly into East Polynesia. And so a colleague and I, looking at the archaeological evidence, the oral traditions, and the genetic evidence, suggested that perhaps what's happening here is about 1500 years ago, when those islands are now habitable. They've had some time to get some soil and so forth, and that we have another movement of people out of island Southeast Asia, and that we might see them visually, archaeologically, on the atolls of Micronesia, and that it's perhaps this secondary migration that is driving that movement out into Central East Polynesia from West Polynesia. So we put that out there. A lot of people thought we were kind of crazy to suggest that, but recently there's been some genetic evidence to support this possibility based on, this is a paper that came out in 2020 in PNAS, looking at over 1,000 whole genome sequences from Samoa. And what they find is evidence of kind of slow population growth, limited population size, and then about 1,000 years ago, 1,200, something like that, they see a massive population increase and then a crash again when Europeans arrive with the introduction of European diseases, and then steady growth from there. So genomic evidence that's supporting the possibility of another population movement in. And in talking about this research, I was giving a seminar over in the biochemistry department at the University of Otago when I first arrived here. And I started chatting with Tony Merriman, who is a colleague who'd been working on metabolic disease, and in particular gout in Pacific populations. And he was intrigued by this suggestion that we have this second movement and not necessarily a direct link between West Polynesia and East Polynesia, because he was finding that a number of the markers associated with metabolic disease and gout, like ABCG2, which is encodes for uric acid transporter, showed differences between East Polynesia, between Maori communities, populations here in Aotearoa, and West Polynesian, again, Pacific populations living here in New Zealand. They have different markers associated with metabolic disease, which affects both populations disproportionately. And when we look at the distribution of metabolic disease, or mean serum urate levels associated with metabolic disease and worldwide, we see the highest rates in the world amongst Taiwanese Aboriginals, East Polynesians, and Micronesians, and then other Pacific people and other communities kind of also suffering from hyperuricemia, but at lower levels. And so we started doing some SNP ancestry chip testing in Pacific populations living here in Aotearoa. And indeed, we are seeing genetic variation between East Polynesian and West Polynesian populations with some of these people here who have mixed Polynesian ancestry. And so it looks like this kind of model that David Addison and I proposed could indeed be going on. So recent research that is also challenging other of these kind of more simplistic aspects of Pacific settlement. In 2016, using ancient DNA, a group out of Harvard and the Max Planck at Jena were able to get whole genome data from skeletal remains associated with the earliest Lapita site in Vanuatu and in Tonga. And they were able to amplify or obtain genome data from 4 crania from the site of Teuma. Now, Teuma, apologize, I should have warned people that I will be showing skeletal remains here. Teuma is a burial site of over 70 individuals, dated to the earliest occupation of Vanuatu, 2,900 years ago. It's a fascinating site. Of all of these burials, not one of them actually has its skull in place. So the crania were removed from every one of these bodies post decomposition. We know that they were not beheaded, that the skulls were removed after the decomposition had occurred. We see teeth, for example, falling out of those skulls while they were still in place. So the teeth are still present there, but the skulls are not. There were only a handful of skulls, all of these in the site. All of these were found in ceremonial contexts. So you can see an individual here does not have their skull, but there are 3 skulls between their legs. There was also a skull placed within a Lapita pot with another Lapita pot plate on top of it. So there's no question that these are associated with Lapita in some way. 2 years later, the same group now split and published 2 papers describing, comparing these ancient DNA results to DNA from later Lapita sites in Vanuatu and other historic and extant modern peoples from Vanuatu. And what they found oh, I probably didn't say, the DNA from those skulls did not turn out to have any, or have a tiny amount only of Near-Oceanic DNA, but were purely island Southeast Asian. But the samples that they processed that post-dated that initial early Lapita sites, so these are still late Lapita skeletons, have the reversed, almost exclusively Near-Oceanic. So the argument was there's complete or near complete population turnover in remote Oceania within a few hundred years of Lapita settlement. But my question has always been, these skulls are in ceremonial context. Do they actually represent the population? We know that ancestor worship and skull shrines are very common historically and even today in parts of the Pacific. And there was evidence on these skulls that they had been curated, they had been managed. So are these potentially ancestors that people carried with them in their waka when they came out? Which is, I suggest, a distinct possibility. Others have suggested that no, they are the same people. But this raises the question now of who were these people who went on to remote Oceania? Are they a mix of Austronesian and Papuan peoples, or do we have exclusively an East Asian population that lived in Near Oceania for several hundred years and not interacting genetically with those people? Or is it more complicated? Basically, it looks like it's much more complicated. And when we look at the whole genomes of these Lapita skulls, the 4 Lapita skulls, these are modern West Polynesian populations, these are modern East Polynesian populations, these are the Near Oceanic and post 2600 BP Vanuatu populations. Things are much more confusing than the simple Lapita story. So most recently, there's been a lot of interest in the Pacific recently. People have applied genomic studies to the peopling, to human genetic studies of Polynesia to try to understand settlement. And sadly, I've been arguing with these people about their assumptions of the likelihood of being able to reconstruct the settlement from modern human populations. But they've argued that they found evidence of a serial founder expansion, directional loss of variance, and they've actually dated the settlement of each of the islands based on the genetic variation of people today. So genome-wide study from 430 modern individuals from 21 Pacific Island populations. So here are the data that they have generated, and you can see here the Central East Polynesian populations, the West Polynesian populations, the Taiwanese Aboriginals, and they have reconstructed the process of settlement, which I have to say we actually already know from archaeological evidence of the dates of earliest settlement throughout the Pacific, and indeed it was very, very rapid. And the separations that we see between West and East Polynesia and between Rapa Nui and these more isolated locations like Easter Island and in the Australs ties in very nicely with what we know about the linguistic differences within Polynesian languages. This is the reconstruction of the dates that these researchers found for the settlement and the directions that they were settled. Fits very, very nicely with the archaeological evidence. However, it doesn't really make a lot of sense when we think about the evidence that we have of continued long-distance trade and exchange. You don't have isolated island populations. We know Pacific people are sailing back and forth. This is evidence of trading basalt, an important stone for making adzes, and people are moving adzes all over Polynesia, from Hawaii down here to central Polynesia. We know that the sweet potato, for example, was dispersed throughout the Polynesian Triangle. So this doesn't fit with isolated island populations. We also know that Polynesia was hit very, very badly by European disease when Europeans arrived, that caused massive crashes in particularly East Polynesia and the communities that these people were studying. So the likelihood that the living descendants are related to, only a handful of those living descendants would be related to those early initial colonists. And we also know that blackbirding and recruitment slave trade took another major hit on Pacific communities. So there are real difficulties with using modern populations to reconstruct these ancient migration patterns using human DNA. And finally, they had very, very small sample sizes. These are legacy samples. They're not working with the communities. And they used an ancestry array that was designed for Latin American populations. We think if you're going to study Pacific populations, there's clearly we've identified genetic variation that doesn't exist in these other model populations. And we need to be targeting Pacific-specific mutations to understand the settlement of the Pacific. So the final, very quickly, things are more complicated. This just came out in July from a study in Micronesia, working on both ancient and modern samples, 164 ancient and 112 modern individuals. David Wright's group, again from Harvard, have identified now multiple, at least 5 migration events impacting Micronesian populations. So Micronesia has been kind of understudied in the past. We now see the complexity when you actually get large sample sizes of both ancient and modern DNA. So things are much, much more complicated than the stories that we developed from the 1980s onwards. And we really do require engaging with that knowledge, that archaeological, linguistic evidence, and most importantly, engaging with the indigenous communities and their traditional knowledge. Chronology matters. We have to include the ancient DNA to really understand the settlement, whether we're looking at human or commensal DNA. And yeah, modern distributions and sequences may not be represented. So it's not simple. We've got to do our homework. We've got to engage. And I will leave it there because I've already gone over time.